everyone, I'm Nicole, and today we're going to be talking about my thesis topic, which is on assessing the role of soil nitrogen content on the interactions between perfluorinated carboxylic acids, or PFCAs, and crops. So PFCAs are a part of a much larger group of chemicals known as PFAS, or per and polyfluoroalkyl substances, which are a group of man-made chemicals that have been used in industry for the past 60 years. PFAS are an ideal industrial chemical as they consist of strong carbon-fluorine bonds that make these pollutants environmentally persistent, chemically and thermally stable, and resistant to degradation under ambient conditions. Due to these properties, PFAS are ubiquitous and are known as forever chemicals. Perfluorooctanoic acid, or PFOA, as seen on the left here, is one chemical included in the PFAS group of chemicals. And as you can see by looking at its structure, the long carbon-fluorine chain is hydrophobic, and the oxygen and hydrogen species on the head group make it hydrophilic. PFAS can bioaccumulate and are known for being water-soluble, as well as having long-range mobility. So PFAS are used in the production of protectant coatings for metals, oil repellents in food packaging, protectants for textiles and carpets, stain repellents in clothes, personal care products, pesticides, firefighting foams, emulsifying agents, and even non-stick cookware. Despite restrictions put in place to ban the use of certain PFAS, many are still used or produced by several countries such as Russia, China, Uganda, South Korea, and India. In 2014, it was estimated that 2,600 to about 21,000 tons of C4 to C14 PFCAs have been emitted globally per year from 1951 to 2016. Future projections state that PFAS production and use will be most likely from the PFOA-based products, and as well as productions of fluoropolymers in China, Russia, and India. So why should we care about PFAS? So PFAS have a mean half-life of about 4.4 years in humans, and it has been observed that exposure to PFAS can lead to things like birth defects, chronic kidney disease, disruption to thyroid mechanisms, cancer, and even could reduce the ability to fight the COVID-19 infection via immune suppression. The environmental and human health impacts of PFOA and PFAS have been well researched, and these chemicals are known for their toxic effects and persistence, and therefore are recognized under the Stockholm Convention as persistent organic pollutants. Other short-chain PFAS are under review. However, there is currently limited research on things like daily intake, toxicity, and persistence to validate this claim. Research suggests that PFAS interacts strongly with proteins and circulates the body through binding to plasma constituents. Specifically, it was found that PFAS have a high affinity to the carrier protein albumin. So PFAS typically enter the environment during the disposal, production, or use of PFAS in processing and manufacturing facilities. Now, PFAS does not undergo degradation under normal environmental conditions, and that therefore can be detected in all biological and environmental matrices such as sediments, groundwater, surface water, air, Arctic ice, soils, terrestrial and aquatic organisms, and even humans. Major contributors to PFAS contamination include the use of firefighting foams, landfills, wastewater effluent, biosolid applications, and using products that contain PFAS. PFAS have also been detected in high concentrations in agricultural soils and crops worldwide. Human consumption of crops grown in contaminated soil is one of the main ways that PFAS can enter the human food chain. It was found in a dietary exposure study that 69% of PFOA and PFHA exposure came from vegetables. Therefore, understanding the relationship between PFAS and the components of agri-ecosystems, such as the interactions between PFAS, crops, and soil, is essential in identifying the risks that PFAS have on humans and can aid in formulating solutions to decrease exposure. Plants generally uptake PFAS from growing in contaminated soil and water. Both active and passive processes have been proposed. Passive processes would mean the uptake of PFAS through their roots and transport it to the shoots and leaves of the transpiration stream via diffusion. And then active processes would be active carrier-mediated transport. However, there are discrepancies in the literature as to the exact uptake mechanism, and this is due to the unique chemical and physical properties of PFAS. It is likely that the uptake mechanism is a combination of passive and active processes, but this will depend on the specific compound, as PFAS vary depending on their chain length and associated lipophilicity. In terms of the mode of action, the toxic response of PFAS on plants is due to oxidative stress through the buildup of reactive oxygen species, or ROS. ROS levels are controlled by dynamic equilibrium provided by plant mechanisms, including enzymatic and non-enzymatic processes, where ROS are scavenged by plant cells. However, under stressful conditions, such as exposure to PFAS, ROS production is significantly increased. So this disturbs this equilibrium as well as the cellular redox state, leading to a buildup of ROS that the plant cannot accommodate for. So this leads to oxidative stress. Exposure to PFAS generally causes cell and tissue damage, but can also alter chlorophyll content as well as photosynthesis, which leads to decreases in biomass. So there are a couple of factors that influence plant uptake of PFAS. The first one is protein content. So protein content changes depending on the plant species. Since PFAS interacts with plant proteins, plants with more protein could accumulate more PFAS. The second one is physiology. So again, different plant species have different uptake abilities and capacities. For example, radishes lack a Casparian strip between the bulb and the shoot, which allows for increased PFAS translocation ability. The next one is PFAS chain length. For example, short chain PFAS are more readily translocated because they are smaller and more hydrophilic. The next one is environmental factors. For example, high temperature and high salinity have been shown to increase PFAS uptake. The last one is organic matter. So research has shown that more organic matter present in the soil allows for increased retention of PFAS. And this is most likely due to PFAS interacting with the proteins in the organic matter. So other potential environmental factors are nitrogen. So studies on artichoke and cabbage found that increasing soil nitrogen content can increase the plant protein content by 26%. Now this may have implications for increased plant uptake of PFAS, but has yet to be studied. So the overall objective of my experiment is to understand the plant response to PFAS in agri-ecosystems and how soil nitrogen content can influence this relationship. So my research questions are, how does chain length affect how plants interact with PFAS? How do different types of plants interact with PFAS? And how does nitrogen affect the relationship between plants and PFAS? So the strategy I will be using is to use metabolomics to see the biochemical interactions as well as overall plant stress response. So we want to compare the effects of short chain versus long chain PFAS, soil nitrogen content in relation to plant uptake of PFAS, and how these differ in lettuce and radish. So overall, we want to determine if plant uptake of PFAS or PFCAs increases when exposed to high soil nitrogen. We also want to determine how plant responds to PFCAs and how that varies between root and leafy vegetables. So the rationale for the nitrogen experiment is that plant uptake of high nitrogen content in soil leads to increased protein content in plants. High protein content then leads to an increase in PFAS accumulation and retention and associated plant stress response. Some evidence of this relationship was seen in my lab when past research was done, and they were looking into amino acid metabolomics. So my first experiment is the nitrogen experiment. So our test species is going to be lettuce. We're going to use 10 replicates per treatment or control. We're going to have a negative control, a positive control, a high nitrogen control, and then 2 treatments with the same amount of PFOA and with high and low nitrogen. So we're going to be using a nitrogen fertilizer, which is a 24-8-16 applied once a week. And then the growing medium will be a potting soil. The next experiment will be chain length. So the test species is going to be radish. We're going to do 10 replicates per treatment or control again. We're going to have a negative control, 2 positive controls, and 2 treatments, one with the same relative concentration of PFAS. And essentially we had to increase the concentration of PFBA simply because there's differences in molarity between PFOA and PFBA. So step 1 is to grow the test species. So we're using black-seeded Simpson lettuce and cherry bell radish because they grow the fastest for each of their species. Seeds will be transferred to germination trays for 2 weeks before we apply any chemicals, and they'll be grown in a growth chamber under the optimal growing conditions based on past research. Step 2 is to apply the PFCA treatments. So that once they are germinated, they'll be put into pots with 1 PFCA per pot. And then the PFCAs we'll be using are PFBA, which is a carbon 4 chain length, and PFOA, which is a carbon 8 chain length. Step 3 is the leaf and taproot extractions. So we will be using a developmental scale to determine when to extract the test species. So once the harvestable mass has reached the developmental goal, they will be harvested and then immediately flash frozen with liquid nitrogen, freeze dried, undergo biphasic extraction, at which point the samples will be ready for NMR analysis. Step 4 is to use nuclear magnetic resonance spectroscopy or NMR-based metabolomics to analyze our samples. So NMR is an analytical platform that is used to measure metabolites. So through using NMR-based metabolomics, I expect to see a shift in the plant metabolome when lettuce and radish are exposed to PFCAs and nitrogen. So this shift will include changes in the molar concentrations of plant metabolites and provide insight into the plant stress response when these crops are exposed to treatments. And step 5 is a PCA analysis, or a principal component analysis, which will be applied to the data set in order to analyze the variation between treatment and control plants. In terms of current results, I don't have any yet, but sample preparation and NMR analysis are in progress. So we expect to see a shift in the plant metabolome and as well as an increase in plant stress response after exposure to high nitrogen and PFCA, signified by an alteration in plant metabolites, which can then tell us what metabolic cycles the PFCAs affect, as well as the level of perturbation from the normal plant cycles will indicate a form of plant stress response. For example, if we saw alterations in citrate, succinate, fumarate, and malate, that would indicate that the PFCAs are affecting the TCA cycle or the tricarboxylic acid cycle. So to our right here is a picture of an NMR spectrum where the peaks represent the entire plant metabolome and where different peaks or collection of peaks represent different chemical structures of the metabolites, as well as their molar concentrations. So this the spectra, it will be very similar to the ones I will generate. Essentially what you can do is compare the treatments to the controls and compare them for different concentrations of metabolites. So in conclusion, PFAS are damaging to both human and environmental health. My research will allow us to further understand the relationship between PFAS and agri-ecosystems and determine if high nitrogen use in agriculture can contribute to higher crop concentrations of PFAS, and also can aid in forming solutions to decrease human PFAS exposure through crop consumption. Future research could be conducted on the impacts of salinity and different organic matter constituents on the relationship between PFAS and crops. Thanks for listening.